My name is Apexa Modi. I'm a molecular biology and biochemistry major, and the title of my research project is the role of the P12 protein during the Murine Leukemia virus's replication. The Murine Leukemia virus is commonly used as a genetic therapy vector. So basically, if a patient has a disease that's caused by a mutation in one of their genes, then one of the final therapies that they can try is gene therapy using viral vectors, such as the Murine Leukemia virus. So basically, they put the correct form of the gene with no mutations into the virus, and then have the virus infect the patient's cells. The first therapy that they tried with this gene therapy method was to cure an immune disorder. About 90% of the patients were cured, but when they did a follow-up 3 to 4 years later, they found that about half of the patients had developed leukemia or cancer. So it was clear that we didn't know enough about the Murine leukemia virus to continue using it safely as a genetic therapy vector. There's a need for a lot of basic science research to understand what the different proteins in the Murine leukemia virus are doing, which is kind of where my project comes in. I'm studying a very small protein in the Murine leukemia virus called P12. What we're trying to do, my project in particular, is trying to figure out what part of P12 is important for this function. And so hopefully by understanding that and understanding what other proteins P12 is possibly interacting with, we might be able to better control the Murine leukemia virus when it's used in future genetic therapy trials. The next steps are to just continue this basic science research, continue defining what P12 is interacting with. It's basically to try to map out what all the different proteins in Murine leukemia virus are doing. And then once we get that full picture, then we can go in and be like, okay, if we modify this protein, then we can do this with the virus. Or if we modify another protein, we can control the virus in a very specific way. So I got involved in research at Rutgers through the Arresti Center. I did the Arresti Summer Science program, where I was paired with a professor who does research on campus. That kind of gave me a great foundation for research, how to go about doing it, how to go about working in a lab and everything. I was really interested in working with viruses, virology, immunology. My professor, the one that I work with, is Dr. Monica Roth, and I just spoke with her. She said she had a few projects that would be of interest to me, and so I chose one and started working with her from there. I've learned a lot of things from doing research. It's definitely made me a better thinker, a better problem solver. Just the way I look at the world now, I can solve problems so much better, so much more methodically, and actually see the small details and things. And then about the research itself, I've learned a lot of details about virology and how much more complicated it is. My advice for students who want to get involved in research is first to ask themselves, what are they really passionate about? What kind of questions are they looking to answer about whatever they're studying in school? And then try to find professors who are kind of doing similar research projects, and then do a little research about the professor's work, and then maybe email them, talk to them, and just be willing to learn and put in the time and effort, and then it should be fine. Usually professors are really receptive to students who want to get involved with research.